The Celgene approach has been to let the science drive us towards the indication. So by understanding how these cells behave, we can, if you will, isolate specific attributes and then use those attributes to treat a very specific mechanism in the disease process. So for example, harnessing the way placental cells probably modulate the maternal immune system, we found and we are very confident that we can use that to modulate the immune system in a patient who has an autoimmune disorder, one where their own immune system is destroying, for example, their cartilage in rheumatoid arthritis, or the lining of the gut in Crohn's disease. One of the interesting things about the placenta that we know is that in women who have multiple sclerosis, pregnancy actually has a therapeutic benefit. These women actually improve during pregnancy. The mechanism is not well understood. We believe it may be related to how these placental cells sort of turn off that autoimmune autoreactivity that destroys the myelin-producing cells of the brain. So one therapeutic approach, obviously, is to administer these cells with the intent of them shutting down this autoreactive destruction of myelin, and we think that that's a very sound therapeutic rationale for using these products. I would say this was 1999, 2000. My research team began to, in essence, deconstruct the placenta, and we found that the placenta was actually probably the richest source of a range of stem and progenitor cells. It had been thought up until then that the predominant cell types would be hematopoietic stem cells that were mostly geared towards developing blood and bone marrow type components. In an organ that was considered a non-fetal or embryonic material, you could recover cells that still had a high degree of differentiation versatility. So all of the placentas that we employ for recovering these cells are procured in conjunction with a very rigorous program of identifying the donors, qualifying the donors, and then recovering the organ under very controlled conditions during labor and delivery. The placentas are returned to our laboratories and then processed using some very proprietary methods in which specific parts of the organ are used to recover different cell types. So before I mentioned that we've got a category of cells called PDACs, placentally derived adherent cells. From that category, we derive very specific subpopulations from which we've developed certain candidate populations for therapeutic products. Our first clinical program has been in the treatment of autoimmune disease in the gut, Crohn's disease, a severe, in many cases life-threatening, but always life-altering disorder where the host's own immune system destroys the gastrointestinal system. The one beautiful thing we've learned about stem cells is they exist in a whole range of compartments, even in the adult body, and by turning on the natural repair or regenerative capabilities of the body, you can actually get the therapeutic end result that you want. The stem cells from the placenta are very, very good at orchestrating and facilitating and driving those activities. And because of that, we believe that the very broadly active nature of placental stem cells will permit us to play in many different disease indications, utilizing different attributes or functions of these cells in different ways. When stem cells first emerged as a potential therapeutic product, there was a lot of concern that we would have to match the donor to the recipient, much the way we do for organ transplantation. That obviously would severely limit the value of these cells to the sort of the broad population of potential patients who might be able to receive them. What we have always focused on, and I think is extremely important, is the placenta is this very unusual immunologic organ. And because when you consider it, the placenta is nature's perfect allograft, right? I mean, the placenta can exist in a maternal host. In fact, in an unrelated maternal host, for example, in surrogate pregnancy, without the maternal immune system rejecting that foreign tissue. That unique biology is something that we work really hard to understand and then preserve those characteristics in the cells so that these cells can be delivered in an allogeneic manner, so that one cell fits all, basically. And this is one of the important distinctions between the placental cell therapy platform and, for example, other sources of cells. Our current model is a true pharmaceutical model of delivering these cell therapy products. So from a single placenta, we can produce many, many thousands of doses of cells. That's important for lots of reasons. Obviously, as we move towards commercializing a product, being able to apply very rigorous quality control standards is going to be essential to meeting the high bar that's been set by the regulatory community, but also in terms of being able to deliver a therapeutic that is reliable, consistent, and economical. Being able to master that culture expansion problem has been a major advance for us at Celgene. When you look at the long-term strategy at Celgene, our approach is understand the fundamental biology of these cells and then apply what we learn to specific diseases like autoimmune disease. Continue to develop the cells as pure regenerative products, namely directing them towards fixing a damaged heart or fixing a damaged brain or whatnot. But the long-term vision must be that a extremely rigorous program of manufacturing and releasing a high-quality product is essential to be able to exploit the broad range of applications.